Hi, I am Mrs. Sloan, and this video is intended for my honors biology students. This is in a series of videos that we are doing right now. I'm going to make myself a little bit smaller. We have already done part one, and part one was the structure of DNA and its replication. And now this is, and I'm looking for a pin here, pointer, here we go. This is going to be part two. And part two, we're going to focus on transcription, which is how you make an RNA message, information from that DNA. And then translation is how you use that message to build protein. All right. So transcription and translation is on our topic today. And if you're new to my videos, the notes my students are using are down in the descriptor of this video, and it also has a link to this presentation within those notes. And column one, I'll help you fill out. And column two, I encourage you to put in pictures that help you with the information that we're going over. All right, so on our notes, this is chapter 24, but we are starting in at 24.2 gene expression. Good to go? All right, so first thing we need to review is the differences between DNA and RNA. So DNA, remember, is deoxyribonucleic acid, and it has a sugar that looks like this, whereas ribose has a sugar that looks like this. And you can see, look at the deoxy part. Here, ribose has an OH, but here, deoxyribose does not. DNA is double-stranded, whereas RNA is single-stranded. And then the other difference is within one of the bases. So DNA has adenine, thymine, guanine, and cytosine. So here's your thymine over here, and that is definitely in DNA. But RNA has the nitrogenous base uracil in place of wherever you'd want to put a thymine in complementary base pairing, you instead would put a uracil. Now, the other thing I want to point out to you is just to remind you about purines and pyrimidines. So purines are double-ringed bases like what you have here with adenine and guanine, and pyrimidines are single-ringed bases. And in complementary base pairing, you always have a purine with a pyrimidine. So on your notes, RNA carries the genetic information. RNA has ribose, okay, instead of deoxyribose. RNA has the base uracil right here instead of thymine, and RNA is a single strand instead of a double strand. And here's just another picture to kind of show you the differences between those. I just like this picture too. All right, so this picture is focusing just on RNA, and you can see I want to remind you what a nucleotide is. It's going to be a base, a sugar, a sugar, and a phosphate group. And here you can see you have uracil. You would never find thymine here. Now, there are three different types of RNA there's actually much more than that, but I'm going to focus on three different main types of RNA and the roles that they play. Okay, all three of these RNA are transcribed from the DNA. All right, so messenger RNA, this is important. It carries the message from the DNA out to the workbench for protein synthesis, which is the ribosome. And these are the genes that are going to get expressed on this ribosome to build a protein. Now, what are proteins made out of? They're made out of amino acids. So transfer RNA is going to carry the amino acids to the workbench where the mRNA is so you can build that protein. And I'll walk you through all of that. So for right now on your notes, mRNA carries the code from the DNA, It's the coding sequence. tRNA carries the amino acid, a non-coding sequence. And then rRNA is the workbench for translation. Now, I want to remind you, when I say rRNA, rRNAs and proteins together make up the ribosome. So here, big ticket items are transcription and translation. Transcription is whenever you use DNA to make any kind of RNA, whether it's mRNA, rRNA, or tRNA. In this case, we're making mRNA, that process, using the DNA to make mRNA. Now, at this point, I'm going to grab a book real quick. All right, so you can think of the DNA as like an entire book of information, right? Our genetic code. But when you make mRNA, you're not copying the whole book. That would be DNA replication. We talked about that in video 1. When you do transcription, you're taking information from just one page in the book and you're building some RNA from that one page. You're not copying the whole book, all right? Then translation is how you use that RNA that you made in order to build a protein. Now, translation, like when you translate something, you're translating it into a different language, right? So that would be like taking English and translating it into Spanish, okay? That's a totally different language. You're taking the nucleic acid language and you're turning it into a protein language, okay? And it's really the proteins that are the workers of our cell, right? Those are the ones that are the enzymes and the structures to make our cells work. So on your notes, on the basic process, number 5, transcription, process by which an RNA molecule is produced. An RNA molecule is produced based on a DNA template, Translation, the mRNA transcript is used to generate a chain of amino acids in a protein. It converts the nucleic acid language into the protein language. Nucleic acid language into the protein language. And that is what is referred to as the central dogma. The DNA is our stored information, okay? We make a complementary mRNA copy of that stored information, and that's the ambassador from the nucleus out to the ribosome, okay? And on the ribosome is our workbench where we actually put together that protein and we do translation, and that is called the central dogma. So central dogma says DNA goes to arrow, RNA goes to a protein, okay? And we can look at exceptions to that later, but that is your central dogma. Now, next thing I need to review with you. Remember when we talked about DNA replication in video 1, we talked about a leading and a lagging strand, right? And the leading strand was so much easier to read because you didn't have to backstitch like you did on the lagging strand. Well, the cool thing is, since you're making RNA and RNA is single-stranded, you can just use the leading strand. You don't need both strands. So the DNA at the region where you want to get that information. Remember, just like a book, I'm not making the whole book. It's like page 75 in that book, right? That region for that protein or proteins that you need, that needs to unzip and unwind. And I only need to use the leading strand. The leading strand on the DNA reads from 3 to 5. I'm reading just like I read left to right. And you make complementary 5 to 3. Just to rewind on that, remember how the DNA strands are anti-parallel. We learned about that last time, running in 2 different directions. So I'm just going to use the leading strand to make my complementary mRNA here. So number 1, underneath transcription, and notice on your notes I said RNA, mRNA, and right here I'm showing you mRNA is produced in the nucleus for eukaryotes. In the nucleus for eukaryotes. Remember, prokaryotes don't have a nucleus. They have a nucleoid region, and so it's not encapsulated within a nuclear envelope. All right? So the DNA first has to unwind and unzip so that you can read the code on the DNA. So who goes about doing that? It's an enzyme called RNA polymerase. Like I'm making more polymers of RNA, just like in DNA replication, we had DNA polymerase. Now we have RNA polymerase. And you need to know where to start. Like if you thought about a jacket, I'm looking to see if I want to bother to put on my jacket over here, right? I can't just put on my jacket and throw the zipper together. It won't connect, right? I have to feed it in and then zip it up. Well, where you feed in your RNA polymerase, where it binds is called the promoter. And controlling the promoter controls a lot about what genes are transcribed, and that's in a later chapter, okay? But for right now, we need to know to start it, we need to first bind onto that promoter. So on your notes, you have number 2, right? Number 1 was DNA unwinds and zips. Number 2, RNA polymerase adds complementary RNA nucleotides to the 3' end of one of the DNA strands. So it's reading 3 to 5, making 5 to 3. And the steps, promoter, start of transcription. So you bind on here, and then after you bound to the promoter, you can start adding in on those nucleotides. So let me show you that in another picture here. Okay. So here we can see RNA polymerase is this big blob right here, and it's bringing in the complementary nucleotides. This is the direction of transcription. It's reading 3 to 5, making complementary 5 to 3. Look up here in the smaller picture, just binding to the promoter. Okay, that's first, that's initiation. And then the RNA polymerase starts reading that gene, making that complementary mRNA, that's elongation. And then you come to a termination sequence and it separates. The RNA polymerase separates from the DNA, and here is your completed RNA. So on your notes, promoter, start of transcription, where RNA polymerase initially binds, to get on there. And then elongation, as the RNA polymerase reads down the DNA template, adding complementary RNA nucleotides to the new chain. Adding complementary RNA nucleotides to the new chain. Okay, now check this picture out, all right? This looks like a Christmas tree or a Hanukkah bush turned, right, if the star was here on top, turned on its side, okay? And you can see like smaller branches and then longer and longer branches as we move to the right. So let me explain what this really is. Running along this center branch here at the very center, that would be the DNA, that center line that you can see kind of in red. Then growing out from this would be your mRNA. So there are literally hundreds of RNA polymerases reading along here, creating these branches, which would be mRNA. So that means once that DNA is unwind and you unzip it and it becomes exposed so RNA polymerase can bind, when one RNA polymerase binds, another one's going to come right behind it, another one's going to become right behind it. And when I look at this picture, because they're shorter here and longer over here, I'm going to assume it started on the left side and they're reading left to right on here, making these mRNA strands simultaneously. Okay, now once you make your mRNA, and this is just showing you one of the strands, look in the background, you can see your DNA, you can see the RNA polymerase, off comes this mRNA. There's a couple things that need to happen to make it a mature mRNA that can be translated out in the ribosome. First of all, you need to protect the head right where it comes off. You put on a cap, okay? And that cap is called 7-methylguanosine, okay? But that's basically just the cap that you put on one end, and that protects because this right here where the cap goes, you're going to eventually start your translation process on that end. The other thing you do is you protect the other end of it, and you put on a tail. That tail is a series of 100 to 200 adenines. So if there's some enzyme out there snipping away at your mRNA, it's got to first snip through 100 to 200 adenines. Okay. Another thing that needs to happen to it is in our DNA, we have things called introns and exons. Now, exon stands for getting expressed, and it's super annoying, but the introns are the part that gets cut out. You can see them getting cut out right here, literally by several different molecules coming together, and you call them SNRPs, and they actually are like scissors that cut out different sections of this transcribed mRNA. And so you have here an exon, an intron, and then another exon. So they're cutting out this portion of it. So when it's all done and it leaves the nucleus, what you're going to have is you're going to have your cap, exon 1, exon 2, and then your poly A tail. And then this can go out and get on a ribosome and get translated. So on your notes, this processing, okay, you have a cap, 7-methylguanosine, placed on the 5' end of the mRNA. Remember, we read 3 to 5, making 5 to 3. So you place it on the 5' end, okay, and then a tail of about 150 to 200 adenines. And then introns are these non-coding sequences. So let me show you right here. Here's your DNA that contains all the exons and the introns. Here is your mRNA. Here you can see the cap and you can see the tail. And then they're cutting out these 2 introns right here and leaving these 3 exons here in the mature RNA. So on your notes, the introns are cut out by a ribozyme, RIB ribozyme. Now, here's something cool about this, all right? Two cool things. The first cool thing is this. Here you left 3 exons, but what if I only left 2 exons? It's the same gene, but by the whatever I leave in or cut out, I can have many different proteins. So this is why your genome is like this big, but your proteome, all the proteins you make, can be of such variety just by how you cut and splice it. My analogy I would give you for that is, remember how I made a copy of that one page in the book, right? Depending on what words I cut out of this paper will then vary in how you read it, right? So just think of all the variations. I could cut out 1 word, 10 words, 50 words, and it'll change the meaning of this protein. So that's the big go-to here on how we have so many varieties of protein from a single gene. Okay. Next, what I wanted to tell you about this was the next thing I wanted to tell you is the ribozyme that cuts this out is RNA based, right? And we think of most enzymes as, and they are protein based, but this is an RNA based enzyme, and that's sort of unique, right? And we'll talk about that when we talk about RNA first world. All right. So on your notes, I think I gave you everything for that. Yay. All right. So now let's try it a little bit and see how we do. So here is a strand of DNA. Now I'm just showing you this. I'm just showing you the nitrogenous base, but I want you to remember that that is connected to a phosphate and a sugar, right? So you're just seeing just the nitrogenous base here. And if we were going to make more DNA, could you tell me the complementary sequence of DNA? And this was in video 1. I know we're in video 2, a little bit of rewind. Could you tell me what bases in the DNA would be complementary to this leading strand right here? Maybe hit pause for a minute, write them down, and then I'll go over the answers with you. You ready? Okay, here we go. All right, so you should have, look how I scribbled it. You should have said, right, C with G, A with T. Why do we have T? Because it's DNA. This would be if we were doing replication, like if we were getting, ready to make sister chromatid, right? T with A, right? A, T, G, C. Hopefully that makes sense. Okay. Now, that's if we were using this DNA to do replication. What if we were making mRNA? What if we were doing transcription? Okay. If we were doing transcription, could you tell me what series of bases you would put here? Okay. Again, I'd hit pause just for a second and try it to see if you can do it. Okay. Good to go. All right. So this would be the series of bases you would have for your mRNA. C with G, that's still the same. Now A with U in the mRNA because we only have uracil, we don't have any thymine. Now notice I have A's here because the DNA had the thymine. The complementary mRNA will contain adenine. Now I want to clear one thing up I hear students say all the time. They say the DNA is changed into RNA. No, the DNA stays the DNA. And if it's a eukaryotic cell, it's in the nucleus. The DNA is not changing. It's still there. You're just using the DNA as a template. So if you have a C in the DNA, you're bringing in a nucleotide G, A with U, T with A, T with A, etc. And you're using this DNA to make your mRNA. You pull your mRNA off, and the DNA will zip back up and go along its merry way. You're not altering the DNA. You're just using the DNA so you know what sequence of bases to put in your mRNA. Now keep in mind, M stands for message because that's what it is. It's getting the message from the DNA. Now, this message is read as a triplet code. Every 3 bases on the mRNA is called a codon. So here's my first codon, GUA, AUC, and CUC. Remember, this is the message. Right now, scientists have figured out if the message says this for a codon, they know what amino acid the DNA is asking for, right? CAT in the DNA is asking ultimately for a specific amino acid because our DNA codes for our series of amino acids in our proteins. So what is CAT asking for? Well, we look up the message GUA, okay, and you use this codon chart. So let's look up GUA. So here's the first row here, G. The second one is U. And look, it doesn't matter. If it's GUU, GUC, GUA, GUG, it all codes for valine. okay? So when the DNA says cat, ultimately the amino acid you're going to get from that is valine. How did we find that out? We looked up the codon, right? And we could look up the next one, AUC. So A is the first one, U is the second one, AUC is isoleucine. That's what that stands for. But now here it's a little bit different, right? Remember here it didn't matter what our third nucleotide is. Here, if our third nucleotide had been AUG, it would have coded for the amino acid methionine. And you know what? This is the only way to get methionine is AUG. This is the one and only way to get methionine. Now, if you look also on here, methionine, though it's not indicated, so it's called the start codon. Every single protein starts off with a methionine. That is the start translating now. As soon as the ribosome is there and you're trying to build it, it has to start with an AUG. Look over here, we have 3 stop codons, UAA, UAG, and UGA. So they don't code for any amino acid. They're like a period at the end of the sentence and says your protein is long enough. So on your notes, you have the genetic code, the sequence of RNA nucleotides that code for our 20 different amino acid. A codon is every 3 bases on the mRNA. That's what you put in on your notes, right? Every 3 bases on the mRNA that codes for a particular amino acid. Every 3 bases on the mRNA that codes for a particular amino acid. And then the codon chart, I gave it to you right here, okay? So I said 1 initiation or start codon, 3 stop codons, and the rest code for various amino acids. All right, so now let's go back. We know what the codons are, okay? And now we know, right, when we looked up GUA, let me go back here. We looked up GUA, it codes for valine, right? So I know what this amino acid is, but who's going to bring it? Who's going to bring it to the ribosome? A tRNA. And the tRNA has to be complementary to the mRNA. So what would be the complementary tRNA? G with C, right? So pause again and see if you can figure out the series here of our tRNA. Ready? Okay. So here, I can see tRNA, each individual tRNA has got a series of nucleotides, but a section of it that is complementary to the codon is called the anticodon. So it has to be G with C, U with A, right? A with U, because we're still RNA, so we don't have any thymine, only uracil. And we know the name of the amino acid it's going to be carrying. Remember, it's T for transfer. Here's where students have trouble. Sometimes they look up the tRNA on the codon chart. You gotta look up the mRNA. So underneath transfer RNA, transfer amino acid to the mRNA that is on the workbench. I'll show you that in just a minute. The anticodon, here's your definition for anticodon for your notes. 3 bases on the tRNA, 3 bases on the tRNA, in this case CAU, that are complementary to a codon, that are complementary to a codon. All right. Who discovered the chart? Nuremberg and Mattei discovered that chart. And I just want to show you, you can get the chart in a lot of different ways. Okay. So you just always look, what's the first base, then the second base, and here they give you the third base. So for instance here, let's look up this codon, CGA. Pause for a minute, look it up, see if you can figure out what amino acid the codon on the mRNA CGA would do. Okay, pause, ready. Okay, here you go. So I'm going to look up C is the first base, right? The second nitrogenous base is G, so I'm going to go all the way over here because the second base, G, so it's going to be, it doesn't even matter what the third base is. Any which way that codes for arginine. Okay, so that's the amino acid that codon would code for. And you should be able to tell me what the DNA was, right? That original DNA that coded for this mRNA, and you should also be able to tell me what the tRNA is from that as well. Okay, so this is just a bigger so first base, second base, third base. I just wanted to point that out to you again. And then just to remind you that this shows the universalness of life on Earth, because all life, okay, we all speak different languages depending on where we live, right? But the life language is universal. It's the same language every single time, whether you're talking about nucleic acids or you're talking about the proteins they code for. Are there exceptions? Yes. I noted a few exceptions on here, slightly different in mitochondria and chloroplasts, and some archaebacteria. All right. But otherwise, the language, all life speaks the same language. All right, so big ticket item here. We've already gone over transcription, how we use the DNA to make our mRNA, rRNA, or tRNA. We've talked about how we process that mRNA. So now translation is how we're going to use that mRNA to build our protein. And we've already kind of got introduced to that. We know every 3 bases on the mRNA is called a codon. We know that tRNA has an anticodon in it that's complementary to the codon. Okay, look way back here. Look at this first mRNA, AUG. Remember what I said? That's always the initiation codon, and that always codes for methionine. That would be the first amino acid here in our growing polypeptide chain. So translation is, how does that all go down? Well, first of all, tRNA is a big part of it, right? So here you can see an amino acid is attached to the 3' end of a tRNA. You can see it has different little loops in this tRNA. It's single-stranded. It's just folded back on itself. Down here at this loop is your anticodon, and it would be complementary to the codon on the mRNA. And this is just another model of it right here. All right, so the ribosome, which is the workbench for protein synthesis, which is made out of rRNAs and proteins. So here you can see it has a large subunit and a smaller subunit, a large and a small ribosomal subunit. And basically what's going to happen is your small ribosomal subunit is going to come first, your mRNA is going to lay down on it, and the first tRNA is going to land. So you're going to have small ribosome, mRNA, first tRNA is going to land on there, and then the large ribosomal unit is going to lock on, and then the light is green. The ribosome is going to read right along that mRNA, and the right tRNAs are going to land, and we're going to start putting together our protein. So that ribosome has RNAs and proteins in the large unit and an RNA and protein in the small unit. And I think, yes, I need to help you with that. On your notes underneath ribosomes and ribosomal RNA, I gave you everything for A, okay, where I explained the subunits. And then B, the function of a ribosome is the workbench for protein synthesis. The workbench for protein synthesis. So here you can see a small ribosomal unit is the dark green, and the larger one is here. And in essence, what's going to happen is these weird tRNA looking things, right? They're going to sit on the ribosome together, and whoever is the newest tRNA to land, he gets dumped on by the older tRNA, and he's going to throw his protein chain on, and the ribosome is going to shift. Then a new guy is going to land, and the chain gets dumped on. And that will keep happening until one of your codons is a stop codon. Now, when the tRNA dumps his, this polypeptide chain onto the new guy, now he doesn't have an amino acid anymore. He's going to need to go and work with an enzyme to put the correct amino acid on the correct tRNA. So when they come, they're delivering the right amino acid to the system. Okay, let's take a look at this picture and see if we can see it, all right? So here is your mRNA running along here, right? You can see part of a small and a large ribosomal subunit. And I'll show you how it initiates. I just want you to see the process. So this guy right here, he's got already 5 amino acids on his back. When this new guy lands, the ribosome is going to shift those proteins. So the ribosome acts as a catalyst, and it'll shift this chain of amino acids onto this new guy, and then this guy will exit, and the ribosome will shift again, okay, until your protein is complete. Now, there are different pieces, different landing sites, 3 landing sites on the ribosome. So I'm going to show you those now. So here you can see here's the small ribosomal subunit. Here's your mRNA. Here's your first codon, AUG. Here's your first tRNA. It's complementary and it's carrying amino acid. Once that's on there, then the large ribosomal subunit will lock on. Now I want you to pay attention. There are 3 sites on the ribosome. Okay. P site, I want to call it P site. It's for peptide or protein. The new guy always lands in the A site. And just think of it this way. He only has one amino acid, the new guy. Okay. And then what's going to happen is the ribosome shifts. And when it shifts, this first tRNA will get moved into the E site for exit. And this is initiation. We've got these pieces all in place, and that's how every protein starts, like this every single time, all life, all right? So on your notes where it says, I already gave you the 3 sites in which they are, so go down to translation requires 3 steps. Initiation, bringing all the components together, right? Bringing all the components together. Small and large ribosomal subunit, mRNA, and the very first tRNA. All right, so now what's going to happen? When a new guy lands here on this, and it's already been going for a while in this picture, here's the new guy is going to land right here in the A site, okay? And then what's going to happen is the ribosome will move, and the ribosome will transfer the chain of amino acids onto the new guy and catalyze that peptide bond between the amino acids. And when it does that, right, this guy's going to land, ribosome's going to shift, then this guy right here that's in the P site, the protein site, he is going to get shifted. Just imagine the ribosome moving from left to right. He's going to get shifted into the exit site. So let's see how that happens right here. And I wrote it up here as well if you need it. So now this guy just got dumped on, and as a result of that, then the one that was here is going to get put in the exit site. See, the CAU is going to be in the exit site, and then he's going to be dismissed, right? And then this opens up the next codon for the next tRNA to land. So this guy that was new is not new anymore, and he's going to dump it onto the next guy. This right here, a series of amino acids, that's your gene expression. That's your phenotype. What was the genotype? It was way back in the DNA inside the nucleus. Your genotype, right, your genome, this right here, series of proteins, that's your phenotype, your gene expression. So this would be elongation. So elongation, polypeptide synthesis occurs as amino acids are added one at a time to the growing chain, okay? And this is going to happen until you reach a stop codon. And at that point, no tRNA is going to land. And when it shifts, it doesn't shift it onto anything. So it releases it, and the small and large ribosomal subunit come apart. And this mRNA could be read by other ribosomes coming right behind it. But now here you have your protein. So that is termination. Components separate from each other as a result of one of the 3 stop codons that we looked at, one of the 3 stop codons. Okay, so putting it all together, okay, here's your DNA. Transcription is making your, look up in the top left-hand corner here, making your mRNA. You're going to put on a cap, and you're going to put on, this would be the cap over here and the tail over here. Also, what has happened is they have cut out the introns, right? So here's your mature mRNA. It's going to hook up with a ribosome, and that ribosome could be on the rough ER, because what makes it rough, right? The ribosomes, and then the proteins could be fed right into the endoplasmic reticulum and then shipped to the Golgi apparatus after that, right? And the first few amino acids of your protein chain determine that protein's destination. Is it going to stay within the cell, or is it going to be secreted? And I gave you that already on your notes, okay? And we'll be watching some videos about this in class. All right, now, what if your message is bad? What if you have bad DNA? What if you have a mutation in your DNA, right? A mutation is a permanent change in the sequence of DNA bases. Now, you can see this play out, right? Wrong DNA, wrong mRNA, wrong amino acids, right? Wrong protein. That's what could happen. So make sure on your notes, a mutation is a change in the sequence of bases within a gene. And this can lead to malfunctioning proteins. So what causes mutations? Well, here's a mutation in a frog, okay? So you could have errors in replication. So somehow when your DNA was replicating, you screwed up, and that's where the mistake happened. Or your DNA could be exposed to mutagens, radiation from the sun, right? That's why you wear sunscreen, right? Cigarette smoke, hello, that's a mutagen. Multiple mutagens in cigarette smoke, and vaping, so don't do it. Okay, and then certain chemicals, okay, are mutagens. Okay, and so errors in replication rare, mutagens, you want to put this in your notes, radiation, UV rays, cigarettes, okay, and certain chemicals. Okay, and then transposons are what are called jumping genes, okay? Literally, these are the most terrifying things to me, and the bulk of your DNA is actually transposons. This is DNA that removes itself and jumps in somewhere else. Okay, evidence of transposons are Indian corn, and this is why. Let me show you. Okay, so here, let me go jump in the Indian corn over here. Okay, here's your normal gene. But if some DNA jumps out of one location and literally excises itself and inserts it somewhere else, it can insert itself in the middle of this gene and mutate it, and now it doesn't have the right series of bases to code for the purple color. Okay, so that is just one piece of evidence for transposons. Okay, what kind of mutations can you have? Okay, this mutation is really bad. This is called a frameshift mutation, and let me explain it this way. Okay, we don't have the genetic code memorized, you and I, right? So I'm going to pretend instead of 3-letter codons, I'm going to give you 3-letter words that you can easily see, right? Can you see the 3-letter words here? Do you see them? Can you read it? All right, so look right here. I separated them out for you like codons. The big fat cat ate the big rat. We can understand that, and it makes sense to us, okay? Now, if you have right here a deletion where you remove the E, okay, in our 3-letter word, we still make 3-letter words regardless if the E is there or not. So instead of THE, then our codon is going to have to be what, right? THB. And you see how the reading frame is shifted. So instead of the big fat cat ate the big rat, it now says Vega ifa attic adatet heba igar at. Do you understand that? No. Every codon is wrong because you deleted one and it shifted it. You could do the same thing by adding in a nucleotide and it would shift the remaining reading frame. So frameshift mutations are when one or more nucleotides is added or deleted from DNA and it causes a shift in the bases of the codon. And this is really bad because every one of these codons is wrong, which means every single amino acid is going to be wrong. All right. Then there's another type of mutation called a point mutation. So you can point right to it. So here is no mutation. ATG. Here's your mRNA, UAC. This codes for this amino acid. We could look it up. Tyrosine, happy face, normal protein. Okay. Now, let's mutate this G into an A. So the DNA reads ATA. So now our codon is changed. Instead of UAC, it's UAU. But lucky, remember how we said there's multiple ways to get some of those amino acids? It still codes for tyrosine. So this is called a silent mutation. It's a mistake, but we get the same amino acid. Our protein folds correctly. Okay. In this one, it has, instead of ATG, it is now ATC. And this is now one of our 3 stop codons. I was trying to model it for you, right? So now you're interrupting that sentence where it shouldn't be, and you have an incomplete protein, and this is called a nonsense mutation, okay? Because we stopped right in the middle of our sentence where we shouldn't have. Okay, and this last point mutation that you're going to see here, instead of ATG, it's GTG, which codes then for CAC, and when we look that up, that codes for histidine. And histidine is a different charge on the amino acid, which causes the folding pattern to be off. So then you have a faulty protein, and that's called a missense mutation, and you see that in sickle cell disease. All right, so that is a point mutation. It involves a substitution of one nucleotide for another, and the results then are variable. Okay, that is video 2. Video 3, which I'll make, will be all about biotechnology. And if you are one of my students, I'll see you in class.